my name is Vishwat. Here, this is my new channel called Bio Theme, which is made for you. Here, I am going to boost your bio learning in various kinds with tips and tricks. This channel is only bio oriented channel. This helps you to ensure that your bio knowledge will be at its peak. So please friends, let's start this new journey with a new way. I will be posting my bio lectures of 11th and 12th standard weekly twice, so please do watch it, and if any queries do comment, I will surely reply. And show some love to this channel by subscribing and like and share. Now let's start the journey. And I have forgotten to tell, and these notes which I'm going to explain every chapter of 11th and 12th is handmade by me when I done my NEET practice. So please guys, make sure that you learn totally perfect. So let's start the journey, and I'm going to start with the 11th standard lesson called as biological classification, which comes under botany, and this is very much important chapter for your NEET exam. So let's start friends. As you see, this is the notes which was made by me during my NEET preparations. So friends, let's start. Yeah, biological classification. Topics under this is comes as Monera, Protista, fungi, virus and prions, mycorrhiza and lichens. Yeah, in this lecture I'm going to explain you about Monera and Protista, and detailed lecture of fungi and virus will be done by me in other sessions. So kindly friends, please do watch all my sessions, it is going to help you a lot. Now basically what we are going to explain in this chapter is, now let us take the topic called Monera, and I'm going to explain about its habitat, about its structure, and about its nutrition and reproduction and classification. So friends, one question compulsory every year in it comes from Monera. The two most importantly from nutrition part, because it has vast nutrition, very vast. Let me rub this, because if you want to make notes you can take through this. Okay friend, let's start. Take the overview of the chapter. This is the way I'm going to start the chapter. Habitat, structure, nutrition, reproduction will be done completely in plant kingdom part, as Monera comes under that algebra for pterophytes. Okay, now let me start. Let me explain. Yeah, now what is Monera? Monera, first going through its habitat. The common question arises that where are monerans found? Yeah, it is diversified around the earth. It is found everywhere, like in soil, water, and air. And these friends take seriously all these points are written in NCERT. And now going on, and even some of the live in unusual habitats such as snow, deserts, thermal springs, salty water, deep oceans. As you know, pressure is very high under deep oceans. Now next, some moneras are also found in guts of animals like methanogens. Remember, discriminate, and some found as parasites. Now friends, as you all know, bacteria is a sole member of Monera. Yes, very important. Bacteria is a sole member of Monera. All member in Monera is prokaryotic and divide fast due to its small size. Yeah, what is the reason that it divide fast into small sizes? Answer is it is prokaryotes. Now next, classification. Our bacteria is classified. It is classified as Archaebacteria and Eubacteria. Yes. Now coming to its structure, yes friends, the coming to structure, yeah, in the structure of bacteria we are mainly discussing about its cell detail of a prokaryotic cell. As commonly we know, the normal cell, what and all it contains a cell envelope, glycocalyx, cell wall, cell membrane, flagella, mesosome, pili, fimbriae, plasmid, nucleus, chromatophore, inclusion bodies, and ribosome. These all the cell organelles which is there in Monerans. Don't worry guys, I'll completely explain all these structures in cell organelles chapter. Don't worry, it is going to come soon. Let me take this off. Yeah. Now next, shape. Yes. Common there are in common there are mainly four shapes called as spherical, rod shape, comma shape, and spiral. Spherical example is very important called as caucus shape. Caucus shape. Now there are three different types in there. This is not there in it. No need to worry about this. This is not there in it. If you want just for reference, you can take it down like two coccus called as Staphylococcus, Chinococcus, Streptococcus, and bunch of coccus is Staphylococcus. Now rod shape, commonly called as bacilli. Plural form is bacilli and singular form is bacillus. Now next, comma shape. Comma shape is nothing but vibrium. It just look like this. Vibrium. Plural vibria. Example of comma shape is cholera. It is mentioned in human health and diseases. Please do take note of this. Very important. Very, very important. Now comes next, guys. What is next? Spiral shape. Spiral shape. Spirillum. Spirillum. Plural shape is spirilla. Example of flagella. In flagella, it is mainly movement shape is spiral. Spiral shape muscles are present. Next comes this your see examples, which is NCERT diagram. Remember guys, very very important. Every year more than 10 to 15 questions comes directly from diagram of NCERT. Never ever leave diagrams. This is cocci, bacilla, flagella, and vibrio. How the question arises, let me teach you about that. Now, as we saw above, cholera is caused by vibrio shaped bacteria. The question comes like, disease cholera is caused by which shape bacteria? And your options A, B, C, D. Then you must take as vibrio. That is answer. Like this question comes in NEET, there won't be any direct questions. They make your mind to search and waste your time. Now guys, next coming to most important part of the chapter, nutrition. Nutrition. So guys, mainly Monera nutritions or autotrophic nutrition. Yes, let me change the color. Mainly it is autotrophic nutrition which convert inorganic into organic, like 6-6-H12O6, which is also called as glucose. Now, what is the process that it convert inorganic to organic? You learn totally in detail about this in photosynthesis chapter. I will be explaining that. No need to worry, no need to worry about this equation too. I'm going to explain this in photosynthesis chapter. Now just take a look of this. First step is there will mainly be two steps like called as light reaction and dark reaction. No need to worry about it. I'll explain this in the next chapter. Now, note. Note is very important, guys. Note is nothing but reduction of carbon dioxide occurs in autotrophic nutrition while by converting inorganic to organic. That's it. simple, and it forms glucose as a byproduct. And anything gets formed, reduction of energy is required. Reduction of energy in the sense it is endothermic process, and means it requires energy. It requires lot amount of energy. That's it. Now next, the two categories of autotrophic nutrition are on the basis of source of energy it is divided. Yeah. Now, on the basis of two sources of energy, what is those names? Those are called as chemoautotrophs and photoautotrophs. Now, as we all know, chemo, the name tells chemical, means what is the sense? Energy from chemical reactions. Where does chemoautotroph gets its energy from? Chemical reactions. It gets its energy from chemical reaction. As we all know, chemochemicals pigments is not present, means chlorophyll, mainly chlorophyll A is not present. The phototrophs, photo, sun, light, yes, energy comes from sunlight. And pigments presents is to capture, as we all know, energy comes from sunlight. It has to prepare its own food, so it has to capture the sunlight. Which pigment captures the sunlight? Mainly chlorophyll A. Remember, I am telling mainly chlorophyll A, very important. Yeah, see, all phototrophic bacteria not have pigments. Yes, 100%, 100%. Yeah, because chemoautotroph is also a type of autotrophs, but it is not have pigment. But here it is having pigment. So the question is, all autotrophic bacteria not have pigments? Yes, true. It is true. Yes, guys. Now, moving on in detail of chemoautotrophs. Yeah, what is chemoautotroph? Mainly the process is like oxidation occurs. Chemoautotroph. What sort of oxidation occurs? What it will release? The release is used to make what? Yeah, outside cell oxidation occurs. Okay, outside the cell oxidation occurs. Yeah. No, see, oxidation occurs as the extends to x naught. Here oxidation is going on like plus one something. Yeah, plus one means oxidation means it is going on to be the sum total plus. And now oxidation cell, outside the cell oxidation occurs. As a result, energy release. The release energy is used to make ATP, which is used for the synthesis of food. During oxidation, which all the elements is used? Which all the elements is oxidized? Example like iron, nitrogen, phosphorus. Very important. These are evolved. Now examples of chemoautotrophs: Nitrosomonas, Nitrococcus, Nitrobacter, and Nitrocystis. Very important. Remember guys, all these are taken directly from NCERT, so don't take literally. Work on that. I'm sure you are gonna get marks from this. Next, yes, here comes another important called as photoautotrophs. Yeah, yep. What is phototrophs? Yeah, as I told you, it requires the sunlight as its energy, and it has pigment, main chlorophyll A. That is our routine use of sunlight pigments present. Don't worry about this. It comes in photosynthesis chapter. I will explain it later. Now, further photoautotrophs are divided into two types: oxygenic and anoxygenic. Oxygenic, as a sense, it produces the oxygen. Donor of hydrogen is water in oxygen. Therefore, byproduct is the oxygen, right? Byproduct will be oxygen. It must be oxygen. Now, anoxygenic. Do not ever confuse between oxygenic and anoxygenic and aerobic and anaerobic. They are totally different. I will explain those in another upcoming chapters. Now, what is anoxygenic? No O2 produced, as name says, an means no O2 is produced. Donor of oxygen is not water. Yeah, 100%. So byproduct will not be oxygen. Simple. And oxygenic, no oxygen is produced. Donor of hydrogen is not water, as in sense no oxygen is produced. Yes. Now moving on. Yeah, this is the overview. This is the overview what I have taught you till now regarding nutrition. Regarding nutrition. Just let me explain you once again. Autotrophs, chemoautotrophs, photoautotrophs. The chemoautotrophs use energy released from chemical reaction, oxidation of inorganic, and recycling of compounds. As I told you, silver, sorry, my bad. Silver, sorry, iron, sulfur, phosphorus, and nitrogen are used in recycling. And next, photoautotrophs. It comes two types: oxygenic and non-oxygenic. The oxygenic source of sunlight, source of energy is sunlight. H2O break and O2 is produced, as I told you, and pigment is present. All oxygenic, all those are oxygenic. As I told you, mainly chlorophyll A and compulsory photosystems are present. I will explain those in photosynthesis chapter, in not to worry regarding photosystems. Now examples are, remember all cyanobacteria simply, like Nostac, Anabina, Oscillatoria, and Spirulina. And coming, remember guys, very much important are examples in every chapter of bio. Remember, example are the key which gives you marks. And I will bring some tips and tricks in the further sessions of the YouTube. So please do be in touch with my channel. Now next comes anoxygenic. As anoxygenic, not produce oxygen. Anoxygenic, not produce oxygen. Examples of purple and green pigments are found. Yes, purple and green pigments are found in this. Yeah, purple and green pigments are found in anoxygenic bacteria. As a result, it can break H2S or some other compounds. Now H2S, in the sense, it is called a sulfur bacteria. And another examples are comes like green bacteria, purple bacteria. Examples are such as green sulfur bacteria, green non-sulfur bacteria, purple sulfur bacteria, purple non-sulfur bacteria. All these all comes under this. Yes, guys. Now coming to the another important part called as heterotrophic nutrition. Now, heterotrophic nutrition mainly saprophytes. It comes under saprophytes, enzyme release outside the body, and complex organic break into simple organic. And see, this diagram explains you. See, simple organic absorption, bacterial enzyme, it complex to simple. It takes on making this cycle. And parasites. Many are parasitic on plant and animal. Example are typhoid, cholera, plague, citrus canker, and tetanus. No need to worry about these three. Mainly these two are given in NCRT. Now another example is symbiotic. Both get benefit. This type of nutrition is shown by bacteria. Rhizobium fixing leguminous. Frankia fix nitrogen in non-leguminous plant called as ulnus. Cyanobacteria also fix nitrogen in colloid root. Very important, guys. Every 2 years once these questions are repeating. Now few observation. See, maximum type of bacteria is heterotrophic. Among heterotrophic, maximum is saprophytic. But the question comes like maximum type of maximum type of bacteria, and option is given as heterotrophic, saprophytic, symbiotic, parasitic. You must mark this as heterotrophic because it is given in NCRT, not this one. Now, simple in structure and complex in metabolic diversity. Different types of nutrition. Yes, obviously, you can see that bacteria are very negligible to visible to naked eyes and even to compound microscope. But you see that simple in structure and it has very metabolic diversity in terms of nutrition. Almost you can see there are 6 types of nutrition, right? Very astonishing. Yes, guys. Yes, guys. Now coming to reproduction, meaning biological process to produce organisms of its own kind is called as reproduction in simple terms. Now the type is divided as sexual and asexual as in common. Now sexual is sexual reproduction occurs, but it is not true. Yeah, sexual reproduction it is not occurring in a true way in bacteria. And Next is asexual. Mainly reproduction asexual. Main reproduction is asexual type of reproduction and require favorable addition like condition, food temperature and pH. Yeah, mainly it require conditions such as favorable food temperature and pH, and mainly fission. It occurs through fission. And during favorable forms spore. Yes, what do bacteria form during unfavorable situations? Yes, it forms spores. Now coming to asexual reproduction, bacteria mainly reproduce by asexual reproduction. Yes, because sexual is not at all true. In favorable condition, like as food is present, pH is normal, it shows fission, binary fission. No, remember guys, binary fission is not mitosis, it is amitosis. Now, see, mesosomes are a type of organ which is found in bacteria. It converts into like a small ball-like structure, and mesosome get double, and it forms two dotted cells where it get divided into half. Now, sexual reproduction in Monera. Now, bacteria, DNA from other bacteria. Yes, sexual reproduction is nothing in the sense recombinant DNA comes and forms bacteria, and the sexual fusion takes place. In bacteria, true sexual reproduction is absent as true gametes, zygote, meiosis, sex organs are absent in bacteria. Remember, no sexual reproduction occurs. Some primitive method of DNA transfer occurs. Some DNA enter into bacteria from outside variation. Yes, this DNA is coming from outside, and the fusion occurs with bacterial DNA and forms new daughter baby or any other mm, further generation products. The features of true sexual reproductions are gametes should form, meiosis must involve, chromosomes should involve, and it should form zygote at the end. These are all features are absent in bacteria, so the true reproduction is absent in the sense. Now, coming to the classifications of bacteria, it is mainly divided into two, such as archaebacteria and eubacteria. Archaebacteria, they are a primitive type of bacteria evolved in harsh conditions, such as examples are methanogens, halophils, and thermosidophils. Now, coming to eubacteria, true, they are called eu in the sense true bacteria. Two or three important groups are present, such as cyanobacteria, mycoplasma, actinomycetes, but they're also called as backed with fungi like future ray fungi. It is actinomycetes is also called as ray fungi, but it's not fungi, it's Moneran kingdom bacteria. Remember, never get confused with that. Yeah, coming to know archaebacteria habitat: high temperature, anaerobic conditions, high salt concentration, high and low pH. It is favorable in all these conditions. You can call it as high salt exosmosis occurs, temperature high, enzyme denature. High salt exosmosis, temperature is high, enzyme denature. Evolution: evolve when Earth have everywhere difficult conditions. Now they are present place where there is harsh conditions. Some difference with eubacteria is NCRT salvo. Salvo is a major difference with eubacteria and archaebacteria, guys. Take a note of that. Now coming to methanogens, halophils and thermosidophils. Yes, methanogens. The meaning of methanogens is oxygen can kill them. In presence of oxygen, methanogens cannot survive at all. Find in it is found in marshy areas, found in gut of ruminants, provides methane. Anaerobic condition occurs. They are obligate anaerobes. Remember, guys, all these are there in NCRT. Examples: methanobacterium. Now, coming to halophils. Halo means salt, guys. Now, meaning the term is halo means high salt areas. It is found near high salt areas like in Dead Sea, where salt lasts up to 2.5 milli concentrations and so on. Then coming to thermosidophiles. Thermo means hot. hot springs in the sense. High temperature where it exceeds up to 80 degrees Celsius, pH is very low, highly acidic as in the sense like 2. To survive high temperature, there are specially made enzymes like a thermostable enzyme. Example like Thermus and Aquaticus. Thermus Aquaticus, not Thermus and Aquaticus. Both are one itself. Thermus Aquaticus. Yes, guys, coming to the last part of this session, it's eubacteria. Eu means true. True bacteria, mainly classified into three types: cyanobacteria, mycoplasma, and actinomycetes. Cyanobacteria, old name is also called as blue-green algae. Remember. Now coming to cyanobacteria, it is found in fresh water and moist soil. Example: spirulina, nostae, cannabina, oscillatoria. Please remember all these examples. Freshwater has the sense even found in marine oceans. Structure: for they are unicellular and it has a mucilaginous covering over that, and sometimes it even form colonial shapes. As now coming to the cell detail, as I told, it has mucilaginous covering. Cell wall is thin and made up of peptidoglycan, which is found only in gram-negative bacteria, and cell membrane is made up of lipid. Cell wall is made up of peptidoglycan. Cell membrane is made up of lipid. Cell wall is made up of peptidoglycan. Cell membrane is made up of lipid. Please remember all these terms. And pigment called as chlorophyll A is present around chromatophores. I'll explain all these organelles in detail in the next chapter. Now, some inclusion bodies called as gas vacuoles, synovian granules are also present. All these details will be taught in the next upcoming chapters. Now, note, cell wall is made up of peptidoglycan, as I told you, and cell membrane is made up of lipid, and it is gram-negative bacteria. Chromatophore is present called as chlorophyll E, and inclusion bodies are present like gas vacuole, synovian granule, and no flagella is present. Remember, no flagella is present as a sense cannot move. Locomotion is absent. No flagella, no movement. Done. And nutrition of that is mainly oxygenic photosynthesis, as in the sense it breaks H2O and releases oxygen. Yes, here comes the diagram of filamentous blue-green algae called as nostoc heterocyst, and it fixed nitrogen. Here are the mucilaginous covering, and this is called heterocyst. Now, coming to, I will explain about heterocyst. Some cyanobacteria cell fix nitrogen. For this, they get modified structure. Nitrogen enzymes develop for to fix nitrogen in them. See here, oxygen is releasing, photosynthesis occurring, and this is called heterocyst. Modified cell nitrogen fixation. Meaning of nitrogen fixation is nothing but N2 gets converted into NH3, nitrogen gets converted into ammonia. Food normal for heterocyst and nitrogen fixed mode heterocyst. Now this enzyme not like oxygen at all, so heterocyst not perform photosynthesis. In the cyanobacteria, it perform photosynthesis, but the cyanobacteria having heterocyst never perform photosynthesis. No, which is present in photosynthesis? The question comes. Examples: pigment, chlorophyll A, nitrogen fixation, and photosynthesis. Answer is nitrogen fixation. Nitrogen fixation is the answer. Examples of these are spirulina, nostae, cannabina, and oscillatoria. Spirulina, previously called as green algae, but it is cyanobacteria. Remember, spirulina is not an algae. It was previously called as green algae, but it is a cyanobacteria. Now, cyanobacteria form blooms in pond water. What in the sense, bloom causes the excess growth and causes the pollution in the oceans, which are lethal to aquatic organisms. Now, guys, next comes mycoplasma. Remember, guys, the smallest creature of bacteria is mycoplasma. It completely lacks cell wall. Remember, completely lacks cell wall. Habitat: saprophytic plus paraphytic on plants and animals. Saprophytic and parasitic on plants and animals. It is free living in soil, can survive without oxygen. It can survive without oxygen. Size: it lasts up to 0.3 millimeter pplo. is the smallest bacteria of mycoplasma which lasts up to 0.1 nanometer. PPL in the sense pleuropneumonia like organisms. PPLO, pleuropneumonia like organisms. Note, cell wall is absent, it is gram negative. Penicillin is not effective. Why penicillin is not effective? Because it completely lacks cell wall. Because it lacks cell wall. This is the answer for this. And shape is not at all fixed and can survive without oxygen, and it has linear shape DNA, linear in the sense straight DNA, linear DNA. Now comes the last, actinomycetes. Look like fungi, but eubacteria. So in the sense, it is also called as ray fungi. See, it looks like this in the tie. It is same as type of fungi looks, but hyphae. It comes in protista. I will explain you regarding those. And bacteria, but look like fungi hyphae. See, look like fungi hyphae. Cannot what is the meaning? See here, it is a bacteria and look like a fungi hyphae. Now you can get the connecting link between fungi and bacteria, as it seems actinomycetes is nothing but evolutionary relationship. It is giving evolutionary relationship, and it is a gram positive bacteria. Cyanobacteria was gram negative. Actinomycetes is gram positive, remember. And coming to examples, Frankia, symbiotic nitrogen fixation. I am remembering you on the time. Nitrogen fixation means converting it to nitrogen to ammonia. I will explain all this in upcoming chapter called as mineral nutrition. And coming to second example is Microbacterium, causes tuberculosis, TB. Third is Streptomyces, genus which produce many antibiotics. Remember, once again I'm going to repeat, Frankia, symbiotic nitrogen fixation into 2NH3. Microbacterium, causes tuberculosis, TB. Streptomyces, genus which produces many antibiotics. Yes guys, I have completed the complete Monera lecture. That's it for today. Please like, share and subscribe, and do comment any queries. I am going to reply each and everything. So please go through this lesson, go through this chapter once again from NCRT after taking this lecture. Yes guys, thank you. See you soon for the next kingdom called as Protista. Bye guys.